And the recording of these proceedings today will be available online. All right, they'll be up within the week. We'll have those available, and we'll have our some of the selected workshops from yesterday posted as well. So just be patient a few days, but all the material will be at our website so that you'll have them as a reference. All right. So I think we're ready to begin. I don't know who's in charge of getting the music turned off for us. We can make that happen.
I'd say dozens.
Okay. All right, there we go. Thank you. It's magic. All right, so we're going to go ahead and begin. We have an absolutely outstanding panel here for us today. Top-notch researchers and clinicians in the field. And in order to facilitate and properly introduce this panel, we're going to have our board secretary and chair of our research committee that everybody knows, Pam Bauer, is going to do the honors for us. And I have to say, Pam has been a real force behind the research activities that have gone on, and she's done so much work in addition to all the other things that you see with her online and all the other things that she does. So it is most appropriate that Pam take the lead in facilitating this session. So Pam, come on up. Thank you.
Good afternoon, everyone. I hope you enjoyed your lunch and you're all ready to sit back and learn all about research. So this afternoon we have a jam-packed program. We're going to give you an overview of research funded by the MSA Coalition over the past 5 years. We're going to look at what's interesting in the MSA treatment pipeline, how you can get involved in research and the importance of brain donation. We're going to end up discussing where the MSA Coalition will focus their research spending over the next few years. It's really a pleasure for me to introduce our keynote speaker, who's joined us all the way from Innsbruck, Austria. He saw his first MSA patient in medical school in 1987, over 30 years ago. And he committed back then to begin working on understanding this disease and hopefully finding a cure. He works harder than anyone else I know to raise the profile of MSA among the research and medical community, having delivered over 300 invited lectures on MSA at medical and scientific conferences. He's published over 600 peer-reviewed articles and book chapters, and has published the first ever medical textbook on multiple system atrophy. He's been a member of our scientific advisory board since 2013, and I'm really happy to announce his recent appointment as chair. Ladies and gentlemen, please welcome Professor Gregor Wenning.
Okay. Pam, thank you very much for this very kind introduction. It's a great pleasure for me to be here. This is the first annual meeting that I participated in, and I'm really overwhelmed by the hospitality and by all the activities that are ongoing here, combining patient needs and scientific progress. So I've been asked to highlight a few of the coalition-funded projects. This is from the SEED program. And then I will finish with a treatment pipeline. These are actually slides that I had also used in the Movement Disorder Congress in Nice just prior to this meeting. So basically, when looking at the projects that have been supported over the year, I think it is impressive that there were 136 applications coming from big sites, big PIs in the field. 36 were selected for support, totaling $1.6 million. As far as I know, this is globally the largest effort coming from a patient support group to fund research, and I think you are all applauded for this very important and continuing support. This is just showing you when the 1 million milestone was passed in 2016, some key members there. These 36 grants went to 25 institutions in 9 countries. It really is a global effort. I think the coalition has matured into a global initiative, and many of the PIs present in this meeting have had the benefit of the coalition support. We will hear about the Global MSA Registry, which is a really important initiative that allows patients to be enrolled. And I think it's about close to 1,000 patients that have been registered here. We will hear about this from Lucy Kaufmann in a minute. So let's go into some of the projects that have been supported. This is just to remind ourselves about the fundamental problem of MSA, which is that of the progression of the disease. It kind of starts at a premotor level when there is involvement of bladder dysfunction, sexual dysfunction, orthostatic hypotension. There may be stridor in some patients as well. This is then followed by a phase that the criteria label possible MSA when there is Parkinson's and cerebellar ataxia. And then when the full house of, or the full manifestations of MSA develop, there are milestones that complicate the presentation. Patients may need a catheter, speech can deteriorate, some patients require gastrostomy and so on. There may be issues with lung infections and so forth. We all know about this, and it's extremely important that we not only focus on the symptomatic therapies that are available to alleviate some of these symptoms, but on trying to modify the disease progression. Disease modification in MSA, that's the magic word, and we need to move towards that. So the first project I would like to highlight was led by Virginia Lee as a PI in Pennsylvania, and based on this project addressing the pathogenesis of MSA, she was awarded a $3 million breakthrough prize in 2020. The grant was awarded to her co-worker Chao Peng. And what did they kind of publish in a very prestigious journal, Nature? It's actually the highest impact journal that you could publish. And so basically, for some time we have known that in MSA brains there is a buildup of synuclein accumulation in glial cells, a bit different to Parkinson's disease, where these inclusions form in the nerve cells. So in MSA, it's the glial cells. And we never quite understood why that is. Why are the glial cells, the support cells in the brain, why do they take up all this synuclein? And in this study that Chao Peng did, under the supervision of Virginia Lee, they established that there are some endogenous factors in glial cells that basically govern the formation of the synuclein inclusions in this cell type. They didn't know quite which factors they are, but they were able to demonstrate that these glial cells have something in common that drives the formation of this toxic synuclein buildup, which is at the root of the problem in this disease MSA. Another project supported by the coalition went to Khurana, who is currently in the board of directors of the MSA coalition. He created a stem cell-based therapeutic platform for MSA, and this was a fundamental work of fundamental importance in MSA treatment strategies. So he was also awarded for this work with the inaugural Bishop Dr. Karl Golser Award, which is globally the only scientific award that goes to research in MSA and PSP, to distinguished scientists. So on that picture you can see Bishop Golser, who suffered atypical Parkinsonism and died from it. And I had the pleasure to accept an invitation to act as the president of this foundation that gives away the award. On the left side is a famous neuropathologist, Gabor Kovacs, who spends a lot of time studying the brains of patients with atypical Parkinsonism. So this is the work that Vic published. And so he is trying to establish the way cells communicate through distinct cellular processes to build up synuclein and trying to identify targets for intervention. Very important work. This is another project by Manu Sharma, who is a PhD. He is an expert in neurogenetics, and he works in the famous Clinical Brain Research Institute, Hertie Stiftung, Hertie Institute in Tübingen in Germany. And he, through a big network involving many investigators active in MSA research, established some shared gene loci between inflammatory bowel disease and MSA risk. So that's an interesting topic that will be followed up in the future. It may indicate that inflammation in the bowel is important not only for Parkinson's disease but also for MSA. Another project that was funded went to Roy Freeman, Harvard Medical School, for his exciting work on cutaneous alpha-synuclein deposition in MSA. So he established that these cutaneous skin biopsy-based synuclein deposits can separate MSA patients from healthy subjects and PD patients, and he received an award by the American Academy of Neurology. And again, this just shows you the type of work on MSA that was selected for support really has fundamental repercussions, not only within the individual research group, but it's actually signposted for further awards, as you can see here, in multiple fashion. So next, the project went to Galbitan in Los Angeles, who developed a new diagnostic test that picks up alpha-synuclein in the blood from MSA patients, and he compared that to PD and healthy controls. And he could actually correctly diagnose MSA with almost 100% accuracy, comparing PD and MSA. As you see on the right side, there are elevated levels in MSA. So basically, it is important to develop biomarkers for MSA, not just relying on clinical scales, and this points us into the right direction. One of the projects went to Innsbruck, where we have a special interest in MSA for many years since I came there in '94, we've seen hundreds of patients with MSA, and more recently we established a gate lab. So basically we are using sensors to develop outcome measures for intervention. And in this study here, using these sensors that we got from a group in Erlangen, which is in Bavaria, in Germany, where they have a lot of experience with this, we were able to separate based on these sensor signatures. While the patients were walking, we were able to separate them from atypical Parkinson's disease. And so I'm happy to announce here today that on the basis of this work, we have now been able to successfully show positive benefits from physiotherapy on gait disorder in MSA. This is a paper by Cecilia Raccagni, who is co-PI here, that is coming out in the journal Parkinsonism and Related Disorder. I would like to close here with a brief summary of these highlights, and I would like now to go into the therapeutic pipeline. What do we have? What is available? And I would like to draw your attention that this kind of pipeline you can actually download from the MSA Coalition website. It's been kind of built with the help of Pam Bauer, who spent a lot of time here. So part of the preclinical drug discovery programs include Exandin 4. This is a grant that was awarded to Vasilius Meissner, who is one of the leading European MSA experts. This is an FDA-approved drug for treating diabetes. And there are many studies, experimental studies, that show that insulin resistance occurs in the brains of MSA patients and may mediate the pathology there. And he has been able to show in this coalition-supported project that Exandin 4 reduces synuclein levels in the brain and ameliorates nerve cell death in MSA mice. This is the next preclinical project where a grant was awarded to Nadia Stefanova in the Innsbruck neurobiology lab, and Gal Bitan again. They looked at a new compound. It's called a molecular tweezer, CLR01, and they found that this molecular tweezer that cuts off certain parts of the synuclein protein and makes it kind of digestible to the phagocytic pathways in the cells, that this can actually reduce the toxic synuclein levels in the mouse brain and reduce behavioral abnormalities. So an interesting approach, and it calls for translational activities with drug companies now. Another study that was funded involved fingolimod. Fingolimod is an FDA-approved drug for treating multiple sclerosis. This grant was awarded to Ruth Paris, and it was this compound was examined in MSA experimental mouse models at the Texas Tech University Health Science Center. So basically, they were able to show that neuroinflammation, which plays an important part in MSA brain pathology, can be reduced. So if you're able to initiate this early on, you may be able to rescue the nerve cells in MSA brains. Now, this is a study or project where a grant was awarded to Eliza Maslaya. I think he is one of the key people in the field of neurodegeneration, based at NIH, where he heads the Alzheimer Parkinson program, and he's always had an interest in MSA. He's published repeated studies there, and in this project, an interesting project, he combined two different approaches to hit the synuclein toxicity. So basically, he combined an alpha-synuclein antibody and an anti-inflammatory drug and showed that he was able to reduce the inflammation, the pathological synuclein accumulation, and he was also able to improve the motor deficits in this mouse model. So maybe in the future, what we might see, what might be necessary, like in oncology, is not to rely only on one therapeutic strategy. You may need to combine 2 or maybe even 3 that hit different targets. This is another study that was supported by a grant to Eliza Maslaya, looking at kallikrein 6. This is an enzyme that cleaves alpha-synuclein in the brain, makes it less toxic. And again, in this project, he was able to show beneficial effects in the MSA mouse model. Next, a compound nilotinib that you may have heard of in relation to Parkinson's disease trials, where it's currently assessed. It's a chemotherapy drug. This project went to Pierre-Olivier Fernagut and Vassilios Meissner in the Bordeaux group. For those of you who don't know this, the French have a national network for MSA, and that's based in Bordeaux. They have several hundreds of patients that they follow there. So they are very strong in MSA research. So basically, this compound is currently tried in PD in a phase 2A, but so far it has failed to show positive effects in the MSA mice. So we kind of don't know how this goes on. We may have to forget about it, but we will see. Now, let me just mention to you some of the trials that have been performed. This was just preclinical experimental work that I have shown you, supported by the coalition. This is the EGCG trial. This is a trial that was conducted in Germany with about 10 centers, recruiting MSA patients on the basis that EGCG as a green tea extract inhibits synuclein buildup in the brains of MSA models. So basically this was a phase 2-3 trial in about 100 patients, and they were not able to detect any effects on disease progression, and there was an issue with liver side effects in some of the high-dose patients. But they were able to show that MRI markers of disease pathology were positively influenced by the treatment with ECGC. Another trial that is currently kind of ongoing by Biohaven involves Vertiperstat. This is the MSTAR trial. Biohaven took over this compound from AstraZeneca. AstraZeneca had done a phase 2 trial with some positive effects in clinical scores. Vertiperstat is a myeloperoxidase inhibitor. So what does it mean? This is an enzyme that increases inflammation in MSA brains, also in Parkinson's disease brains. So if you inhibit this enzyme, you can deliver this compound orally, so it's a pill that you have to take. You can reduce inflammation in the brain, and if you are lucky, you can rescue nerve cells. On the basis of the phase 2 trial that was done by Astra, a phase 3 trial program is currently in progress in the US and in Europe. And this will be the largest ever MSA trial performed in the history. So in terms of sheer numbers that are involved. And it's an activity at the global level. Now, I just want to mention a couple of small studies because they can also be helpful to open your eyes, you know, what may be useful in the future. Both of them are by Dr. Novak. This is a study using immunoglobulins, intravenous infusions, again to reduce neuroinflammation. And the results were that this administration was safe in MSA patients and well tolerated, but we don't have any convincing data on clinical effects of this. This is another one published by Dr. Novak with intranasal insulin. Insulin has vasodilatory and neurotrophic effects. And a phase 2 trial in PD and MSA subjects was recently completed by Dr. Novak. The conclusion is, again, this is safe and tolerated well, but we don't have any convincing data on therapeutic effects. This study here, Sirolimus, is a phase 2 trial by the New York University group, Horatio Kaufmann, Lucy Kaufmann, Alberto Palma. It's an immunosuppressant. Rapamycin is an immunosuppressant used in oncology that enhances autophagy. That means it enhances the degradation of the toxic synuclein inclusions. It rescues nerve cells, but it doesn't seem to affect motor behavior beneficially in MSA mice. It reduces synuclein aggregates, and so this phase 2 trial is currently ongoing. As far as Horatio told me, recruitment is pretty good, and they will definitely be able to answer the question whether there are any promising signals with this. Now let me mention the Coenzyme Q10 story. Most of you probably know that MSA is a sporadic disease. It seems that we don't have any links from, or clues from the genetic field. That may change though. In Japan, 6 years ago, they discovered the first mutation in some small families that can actually cause MSA in that population. So these are very small families, autosomal recessive, so usually brothers and sisters affected. But they had a clear link that these mutations in the coenzyme Q2 gene cause MSA. So the result of that was that the levels of coenzyme Q10, which is at the end of that cascade, are reduced. Coenzyme Q10 is an important part of the mitochondrial respiratory chain. So if you don't have coenzyme Q10 levels sufficient in your brain, that causes neurodegeneration. So on the basis of that, we also have some studies in Parkinson's disease using coenzyme Q10. On the basis of that, the Japanese investigators decided to go ahead with a phase 2 clinical trial of coenzyme Q10 in MSA of the cerebellar presentation, which is the more common one in Japan. So this is quite exciting, I think. This is ongoing. Another exciting development is the immunization. There is the possibility to immunize the body against synuclein, stimulating antibody formation. These antibodies against synuclein then enter or pass the blood-brain barrier, and they may be able to stimulate the removal of synuclein aggregates in the brain. So basically, together with some clinical sites in Europe and a company in Vienna called Apheris, there were 2 trials initiated, one in Parkinson's disease and one in MSA, a smaller one in MSA. And it seems that it is possible to stimulate antibody production by this approach, which is subcutaneous injection of the epitope, so synuclein deposits subcutaneously stimulate synuclein antibodies. So it's a safe procedure. You can actually get detectable synuclein antibodies. The phase 1 trial in MSA was positive, and the phase 2 trial, there is a discussion with the company whether that goes on or not. A decision has not been made yet. I would like to mention this study here, mesenchymal stem cells. You probably heard that in Korea 6 years ago there was the only positive trial in MSA published using mesenchymal stem cells that were infused into the arteries and intravenously, and they were able to show that patients who had that stem cell treatment didn't progress so rapidly compared to patients that didn't receive, that just received saline injections. So on the basis of this study back in 2012, the Mayo Clinic decided to go ahead with a small phase 1 safety trial to check whether these mesenchymal stem cells, when delivered intrathecally, so not into the vessels, but by lumbar puncture. So these patients got a lumbar puncture in the Mayo Clinic, and they had these stem cells administered, whether that's safe and tolerated. And this has just been recently reported in the journal Neurology. It seems to be safe, and they had some evidence that clinical markers were positively influenced. The only issue that they reported that might be of concern, these lumbar punctures can lead to local inflammation due to the cells that are injected there, and some of the patients had pain, like lumbar pain or shooting radicular pain. So this is really something that we need to look out for. Currently, the Mayo Clinic people have filed for a big proposal to establish for sure whether stem cells are working or not. So this is something ongoing. Another intervention I want to mention here, I'm almost done, I'm not sure how I'm doing with time, is PBT434. It's a compound that works both on iron and synuclein. There is a lot of iron in MSA brains, and iron can be toxic. So basically, it makes sense, to address the issue of iron toxicity in MSA. So with this compound, you have a double hit. It can reduce toxic iron buildup, and by doing so, it can reduce synuclein as well. A phase 1 trial has recently been completed, and there were no issues regarding safety and tolerability. One other compound, and that could be a magic compound, that's ANLI-138B, and we have been involved in Innsbruck in the development of this closely. This is a compound that was discovered by a Munich group, by neuropathologists in Munich, Professor Giese. He found this compound, and he was able to show that ANLI-138B stops synuclein from building up in brains, in brain models of Parkinson's disease, MSA, that's worked together with us, and also prion disease. It seems to be a global, if you want, Mr. Proper. I don't know whether you have that here. What's that? Mr. Clean. So in Europe, it's called Mr. Proper, so Mr. Clean. So it really stops the buildup of aggregated synuclein, and we have very powerful preclinical data that it works in MSA models. So I'm pleased to announce here that money has been collected. They started a small company, and they have close to $20 million now to stage a phase 1-2 program with this compound. I'm very excited about this. This is my summary slide that just shows you on the x-axis different compounds in their different phases, preclinical phase 1, phase 2, phase 3, or on the market, and y-axis is levels of success so far. Is it promising or maybe not so promising? And as you can see here, on the top I put ANLI-13B. I don't know whether you can see that. That's the second circle in blue on the top, on the right. So very promising preclinical data, and we need to go into phase 1 and see how that works out. I told you all about the EGCG story, the green tea, that's kind of phase 3 down below, where we didn't have a clinical impact, but at least we were able to influence MRI changes in the MSA brains. Let me just finish this here by saying that there are also initiatives to work on synuclein antibodies. I mentioned immunization against synuclein, that's active immunization, but there are also programs on passive immunization referring to alpha-synuclein antibodies. So there is some data out there as well. So as you can see, when I started 25 years ago, I would have presented you just zero. There would have been nothing. And now you have a long list of compounds under development, and I think that list wouldn't be there if you hadn't supported this program with all the different seed projects, and Khurana is going to tell you about the future there. I think we are in a position now to crack MSA if we follow the right strategies, and if we listen to people like Vic or Daniel or anybody here in the field interested in interventional therapies. I've been asked to talk a little bit about cautions regarding stem cell trials. So FDA, they they are very critical about this now. If there's no definite development program, they don't want to, mention this or stop these interventions even. Google plans to ban ads for stem cell therapies. I just want to remind you, when the Koreans did the stem cell study in 2012, I was flooded with email requests by European MSA patients, where can I get this treatment, and I want to get it done. And then I usually told them, well, it will cost you $30,000. You can get it maybe in China or India, but I would not really go for it if it's outside a scientific protocol. But these patients still went there, being desperate and so on. And I think it's necessary for us clinicians or clinician investigators to highlight the potential dangers. If this is offered outside of scientific protocols, it should not be pursued. So basically, there's also advice. If you're considering treatment in the US, FDA has reviewed all these sites. You can get information. If you go to other countries, as I mentioned before, I would really stay clear of that. So this is where we go. As I mentioned before, in the middle, this was all absent 25 years ago. There was no hope for nothing. And now we have big hopes, and I think it's important for us as clinicians who know about the disease and scientists who've been working on these preclinical screens, that we get together with the drug industry and develop the best possible protocols that give us the best answers for the problems. So with that, I would highlight the Innsbruck MSA group. We've recently started a Dysautonomia center. I want to acknowledge the support over the years by the MSA coalition, but there's also a lot of infrastructure support, both by the university in Innsbruck and the hospital administration. Thank you. Okay.
Thank you so much, Gregor. That was fantastic. So the next speaker will be Dr. Daniel Claassen. Dr. Claassen is a neurologist and MSA Coalition board member who cares for patients with MSA as part of an interdisciplinary team at Vanderbilt. He works closely with his colleagues in autonomic medicine to determine effective neuroimaging approaches, biomarkers, and clinical findings. He has a personal connection to MSA where one of his family members suffers from the disease, so he knows the realities of this affliction. As a clinical researcher, he is also well equipped to discuss what is involved in clinical research for MSA patients and family members. He's going to discuss for us how to get involved with research. Thank you, Daniel. Please welcome Dr. Daniel Claassen.
Can you see?
I'm sure the suspense is killing you. Here it comes, I think. Okay. Well, it's good to be here. It's good to see familiar faces and meet new people. So I'm going to not be too scientific. I know you guys have just been bombarded with very interesting scientific principles, and I'm going to be very practical. And I'm going to give you a 12-minute rundown of what happens in a clinical trial and try and get you excited to get involved in clinical trials and get your friends to be involved in clinical trials and get with your centers where you're getting taken care of to get your physicians involved in clinical trials. All right, so I have two boys. Their ages are 11 and 8. And this morning at about 6 o'clock, my 8-year-old really thought that there was some kind of complex spy thing going on in the Magic Kingdom. I don't know where he got it from, but that's what he thought. And he's convinced that there are secrets in the Magic Kingdom that he wants to find. So he found a website and started reading about it. So it turns out there's some interesting things. So we're going to talk about clinical trial secrets because we're at the Magic Kingdom. All right? All right, so I take care of a lot of patients and we do a lot of clinical trials. I think the important thing to remember about clinical trials is that we need them if we're going to find better ways to prevent, to slow progression, to diagnose or treat disease. In this case, we need clinical trials in MSA. We need lots of different types of clinical trials in MSA. It's a rare disease. There's a lot of variability to it. There are a lot of different things that can cause progression to accelerate in certain people, so we need them. The critical point that I meet with my patients with is I say that clinical trials are not therapies. They're not effective treatments. And I think that's one of the big things that generally people wrestle with. If I don't get in a clinical trial, that means I'm not getting a life-saving treatment. Or if I get in a clinical trial, it means I might be cured of my disease. They're not always like that. We need to do these to find out the answers before we can actually get effective treatments. Clinical trials happen in places where there's clinical medicine. So usually, in the case of MSA, which is a rare disease, they usually happen in big medical centers. So we're hosted by Dr. McFarland. He's got a big MSA center in the University of Florida. He gets involved in a lot of clinical trials. And if you wanted to go check out University of Florida, you weren't there and you live nearby, you may want to go there to find out about clinical trials. Where I practice at Vanderbilt, we take care of a lot of patients in the Middle Tennessee and surrounding regions. We have a lot of people coming into our clinic and interested in clinical trials. Generally speaking, you find a big major academic center, you find clinical trials. However, you can also find clinical trials in clinics that aren't linked to academic centers. A lot of clinics now, like some clinics here in Orlando even, have opportunities to get involved in clinical trials. You don't have to be involved in a big, large academic medical center. And even there are some clinical research sites in the United States that have clinical trials. So they can happen in a lot of places. The key part a lot of people ask me about is how do I find out where a clinical trial is? And I think generally speaking, your best bet is to look at this website called clinicaltrials.gov. Now this is a website that kind of maintains all the current recruiting and even the past finished clinical trials. And in the case of MSA, you can go to this website and you can type in multiple systems atrophy and press enter, and you'll get a huge list of clinical trials that are going on in the United States, around the world, to get an understanding of what people are doing and what's recruiting, what may be interesting for you, what may be not so interesting for you. You can also talk to us at the MSA Coalition. We try and keep up an active list of trials. We try and help companies advertise their trials so that patients who are newly diagnosed or asking about MSA can get connections. It's also important to talk to your physician. Your physician should be able to also navigate sites like clinicaltrials.gov for you and tell you, oh yeah, I know that person in Minnesota, or I know that person in Tennessee or Florida, and I can call them for you and find out what's going on. So it's really a dialogue and kind of updating your information to find out where trials are. So here's an example this morning. I typed in multiple systems atrophy, and I pressed enter in clinicaltrials.gov, and I said, I want you just to show me trials that are recruiting. So here's one, I circled it for you. So it's a study of BHV3241, that rolls off your tongue nicely, in patients with MSA. We've already heard a little bit about this study from Gregor, but essentially you'll see here that its condition is MSA, the intervention is a drug, and then a placebo, and then the locations, you can actually see which sites are doing this study, and you actually can kind of go through and see if that site's close to you or there's a way you can get to it. And so this is an example of one that's currently recruiting, and you can get on the site and find out if it's good for you. So what are the types of clinical trials? And I think this is important as you think about getting involved in clinical trials, right? So there's observational trials, which are really important for us to get a sense as to how the disease is progressing, what are the clinical symptoms people are struggling with, what's important to patients, what's important to your health that you think needs to be addressed clinically. Generally speaking, these observational trials don't require any therapeutic intervention. That is, you don't get a drug or a placebo, you don't get a treatment, you just are followed clinically. There are phase 1 trials, and these generally are first in human studies. So this is where you have a compound, perhaps you have done some toxicity studies in mice and primates, and you feel pretty comfortable that there's no issues that would be given in humans, but you really need to find out. And so you would do a very structured and careful study where you give a dose of that therapy to an individual, and you make sure there's no clinical problems or safety issues that arise from giving that medication. A lot of times, what happens when you do those phase 1 studies is that you might give that therapy to a patient and follow them for a long time and to see if there's actually a clinical effect. And so in phase 2 studies, which generally occur right after phase 1, sometimes they're kind of blended together. We call them phase 1B, 2A, all these numbers. But essentially the idea is what I'm giving you, is it hitting the target? Is it causing a possible clinical effect? And if so, then I may go to a larger study to ask the question, is this clinically relevant? Does this therapy actually do what I think it's going to do? And then sometimes you can have what's called open label, where once you finish, say, a phase 3 study, you're guaranteed to get to clinical study. So you may, for 20 weeks, get placebo or treatment. You don't know what it is, but at 21 weeks, you're definitely going to get the treatment. So that's kind of the overview of the kind of trials that you might come across when you look at MSA. So my biggest advice to you, if you're wanting to do clinical trials, is know what you want to get into. Know what's going to happen. And this is information that's completely open to you. You don't have to search for it. It shouldn't be hard to understand. It should be very explicit to you. And we use what's called an informed consent to go through the entire trial with you. So these questions that could come up. So, how long is this going to take? What do I have to do? What exactly is this thing that you're giving me? How are you giving it to me? What are the procedures that's going to happen? What is the risk? All these kind of questions happen in the informed consent. So as a patient and as a caregiver, this is a really critical part of clinical trials that really puts the power to you. You are the decider of whether or not you want to do or not want to participate in this study. And it's important to go through the informed consent and make sure you understand it, because if you don't understand it, then it's not a very good informed consent, then the communication is not going very well. So inclusion criteria are referred to when the patient, what characteristics define the patients that are for that study. Not every study is for everybody. Some studies you may be a good person for, some studies you may not be a good person for. But we generally talk about inclusion criteria to define what's the study that really fits you as the individual. What characteristics do we want to look for in this study? And sometimes we also have what's called exclusion criteria. And so this can sometimes be a downer. This is my family on one of the log rides, and you can see my wife is a little bit petrified in the middle. But sometimes, there may be things that happen in your life or medical history that make you not an ideal candidate for the study. And that's not a bad thing. It doesn't mean that you failed or anything like that. It's just part of the study, because you want to make sure that the risks are appropriate, that there's no issues involved in this study. And so we talk about exclusion criteria to make sure there's nothing that you have or there's nothing that's happened to you that makes sure you may not be the right fit for the study. So what we try and do in clinics is we try and do what's called pre-screening. So, what's better than one ba? Two bas, right? So we'll look at your information very carefully before, gosh, you guys are slow. It's like getting laughed 5 seconds later. So we'll look at your clinical information maybe before you even come in the screening, just to make sure there's no surprises. For instance, if one of the studies requires you to have an MRI scan, then having like metal in your eyes or your neck may not be a good thing for an MRI scan. So we may look at that in your chart to make sure that you don't have to go through the whole screening and all those procedures to make sure you're not eligible. So who are the people you're going to meet during a clinical trial? Your number one person that you're going to meet is going to be a clinical trial coordinator. Now this might be a research nurse. This may be someone who is like what they call a research associate. But this person is going to be your main go-to person. At our clinic, you have their cell phone number. And so you'll text them when anything happens, or if you have any questions, you can call them. This is your main kind of conduit to that center, that medical center where you're getting that treatment. You'll have a clinician involved. That clinician will probably do clinical exams or talk with you about the treatments. They may even deliver the treatment. Sometimes we do treatments where we do lumbar punctures. And you'll have ancillary staff that you'll meet. So these are the kind of things that might happen in a trial. So you may get an MRI scan of the brain. This causes some consternation with patients, because if you look at that bore, it's kind of small and people get a little claustrophobic. But that's MRI scan is really one of the ways we can non-invasively look at what's inside the skull and see what's going on. So sometimes if that's a concern to you, you can do things like a mock scanner. So our institution will have a pretend MRI scan. You kind of get into it, get used to it, make sure it's not scary, things like that. But that's a really important procedure that happens. Sometimes you do what's called lumbar punctures. That sounds really scary. But this is a picture of my friend Ed Wild, who does a lot of work for Huntington's disease, and he's done now 4 lumbar punctures himself, of his own lumbar puncture, to show people that it's not as bad as it sounds. In that procedure, you may kind of sit on the side of a bed, you may get some numbing medicine in your back, they put a needle in your back, kind of like an epidural, if you were pregnant and had an epidural, those kind of things, and they take fluid out. Sometimes you have what we call cognitive testing, and that's a word that makes you think, oh gosh, I'm going to fail or pass. But here's what we're looking at is how fast you think, how well you think. In this example here, someone's got a list of words and they had to kind of list as many words as they could, and they could write it down. And that's something that may happen in this. So you have a lot of different procedures. Some are invasive, like a lumbar puncture. Some are sometimes difficult to deal with, like an MRI scan. And some are pretty straightforward and just conversation. You take notes, kind of like a neuropsych test. So what do you get from participating in a clinical trial? I think the big thing is you help others. I think so many patients with MSA are so selfless, and they really want to help others. They want to help the next generation. They want to help people with MSA. And I think clinical trials is a great way that you can really invest your life in the lives of others and in the future of science. I think it's really fun to be involved. We find that our patients love to meet with us. We often meet with them like every 4 weeks. We get to know them really well. They get to know us very well. We share stories about outside of work. It's really good to be involved. It's fun to test treatments. It's fun to be involved in the clinical science progress to see if these treatments are actually helping or not. And I think the biggest part that we try and emphasize to our patients is that we want you to be part of our team. We work with you. We want you to tell us what's important to understand, what's important to do, how things are working. And I think that's really the best part, in my opinion, of being in a clinical trial, is that you get to be part of the team. So, what do you want to do now? Get involved. Talk to your clinicians about being involved in clinical trials. If they're not involved in clinical trials, ask them why not. And then if they say, well, they're not interested, then say, well, tell me who is, because I want to be a part of it. Tell your friends to get involved. You'd be amazed how many people we get through clinical trials through word of mouth, or through Facebook, or through other social media entities. Empower your clinical team. We've had so much success in having patients and caregivers help our team become better at what we do. And stay in touch. Stay in touch with the MSA Coalition. Ask us questions. We're here to help you get involved in trials. So thank you, and these are my boys, and they're right now at Epcot, but I look forward to talking to you more. Thanks.
Thank you very much, Daniel. Our next speaker, Dr. Lucy Norcliffe-Kaufmann, is an associate professor of New York University School of Medicine. She's a founding member of the Autonomic Disorders Consortium and an investigator in the natural history study of MSA. She's also investigating new therapies for MSA and developing both disease-modifying and symptomatic treatments. Her research work has been funded by the MSA Coalition, the NIH, the FDA, and the Michael J. Fox Foundation. Please help me welcome Dr. Lucy Norcliffe-Kaufmann.
Okay, thank you very much. Can you hear me okay? It's a real honor to be back here at the MSA Coalition. It's wonderful to see such a growing community of support and advocacy and activities going on. And I want you all to know that that is incredibly important to our ability to bring new therapies to patients with MSA and their families. We've heard fantastic talks this morning, and I hope you are all very encouraged that there are new treatments going on. There is a research infrastructure that exists. Now, I'm going to take a step back from all that and tell you while that important work in developing therapies is going on, we're also doing equally important work observing the characteristics of patients with MSA over time to help support the development of new therapies. I'm incredibly grateful to Coalition who have supported this project almost since its inception, and I hope you will see what an important role that the natural history study and the MSA Registry play in bringing new therapies to patients with MSA. So where are we now? We have a population of MSA patients. We need them to feel better, and we need them to also progress at less fast rates or cure the disease. And to do that, we need drug development. You saw that long list of compounds that we're all developing, we're thinking about. But in order for that to actually make it to the clinic, we need to partner with academic clinicians. We need partners in industry that are going to help us bring those therapies in front of the FDA for approval so that you can obtain them. And we also need to partner with advocacy. So with this network, this will enable us to have a true pipeline for new therapies to enter the clinic. So we are moving forwards, and clearly you see that there's a lot of activities going on, and there are several reasons why we need to be optimistic. The availability of several potential therapies to prevent, delay MSA is an important advancement in the field. But the future success of all of these clinical trials, no matter which compound we choose and which compound actually works, will depend on us having high-quality natural history data and sensitive ways to measure changes in patients that might include various different things, from biomarkers to clinical tests to rating scales to wearable technology. But we need to do our homework if we're going to be ready for clinical trials to happen. So at the natural history study and the registry is really a collaborative effort between government agencies, between advocacy groups like the MSA Coalition and the Michael J. Fox Foundation, and also industry supporters like Theravance. So with that, we have, they're collaborating together as part of the Autonomic Disorders Consortium. And within the Autonomic Disorders Consortium, we have this contact registry for multiple system atrophy, and we have a natural history study in which we're tracking patients with MSA over time at their regular clinical visits, and sites that we're working with are sending us that data, and we're using that data to understand the disease. So today, I'm really going to focus very heavily on what is the natural history study and why is that important for you. So a natural history study, as you heard from Dr. Claassen, is an observational study. It's not a clinical trial in which you're given an intervention. It follows the evolution of patients over time. We know that multiple system atrophy is a problem with this protein that attacks the glial cells and spreads throughout the nervous system, destroying the cells. We're aware of that. We understand what we think is the target of and the core process that causes MSA. That leads to slight difficulties in the beginning phases or problems with autonomic function, and eventually progresses to the point at which people need wheelchairs and eventual death. Right now, we can diagnose MSA, and you've given me this information, or given us, the community, this information through the GLOMSAR registries. But it takes us around 3.5 years since the first symptom that doesn't reach diagnostic criteria to actually reaching that clinical diagnosis so far. So we have a big window of opportunity in which we can intervene, but we can't yet pick up MSA in those stages. We need to be able to pick up MSA sooner so that we have a chance of intervening at an earlier time so that we can save function. So if we improve diagnostic accuracy at this early stage of the disease, we are enabling ourselves that chance. So we need ways also, in addition to picking up MSA sooner, we need ways in which we can measure the evolution of patients and help us think about what are the best ways to do clinical trials, what are the best designs we need. And in order for us to do that, we need biomarkers. We need things like an image bank, right, where we put all the MRIs together and we use that as a resource to develop what is it we should be doing, what is it we should be measuring, what sequences do we need for patients. And we also need that all-important brain bank that you're going to hear about later. So let me move on and tell you a little bit about specifically the natural history study that I'm very involved in. I'm going to give you a brief history of the project. I'm going to talk about the current goals of the project, how the data is being used, and what future directions we have. So this is a timeline of the project, and you can see that it's really a decade of work, right? So this is a long, ongoing project, and it's involving multiple people. The first patient was enrolled in the natural history study in 2010. In 2013, we expanded the study to be able to, with Dr. Gregor Wenning, to be able to collaborate with the European MSA study group, and that was possible with the partnership of the MSA Coalition, and you'll see what an impact those partnerships had on this project. We got renewed funding from the NIH in 2015, and we're continuing this project over time with stakeholders in industry who we also believe play an important role in this project, and we hope to get many more industry partners involved. So the natural history study of MSA, here's some big highlights of this. It's a decade worth of work, right? It's an international collaborative effort. We have 22 sites across the world that are actively participating and sending information from their MSA patients to this database. It consists of this network of highly trained investigators from across the world, and in doing so, it brings together expertise in different areas that are all affected by MSA. We have experts in movement disorders, we have experts in autonomic, experts in sleep, and experts in neurology, biomarkers, imaging. We want those people, and those people are involved in it. So those people are coming together under the umbrella of this project to help support the development of new therapies for MSA. is really exciting. It's a prospective study design, meaning we're going forward and tracking people. We're using pooled data, and in doing so, we're sharing our resources. So what we learn in our clinic can be shared with other clinicians and other investigative teams outside in our network. And it's very focused on MSA. So it follows people with MSA from the early stages. Remember I told you that we needed to be able to pick up MSA sooner so we could intervene sooner. We're catching MSA patients in the pre-motor phase, and I spoke a lot last year about how we have refined our earlier diagnosis of MSA. We're following those people until they develop problems with movement or some of the other CNS problems that patients with MSA, the brain problems, the central nervous system problems that patients with MSA have. We're following into the phases when the movement becomes substantially affected, and we're also following patients up until death and hoping to collect pathology, which is a delicate subject, but it's very important to us. So you can see that we're capturing MSA either from the stage at which people present with only autonomic problems or problems with acting out dreams at night. We're following them in the motor phases, and we're also capturing a small group of patients that have Parkinson's disease or dementia with Lewy bodies that are kind of like our active control group, because we need a comparison group to be able to look at MSA in parallel with. So the goals of the project, what we set out are ambitious, but they are important. We want to support the development of safe, new, effective therapies for people with MSA. That includes therapies that are disease-modifying, that halt the progression of the disease, but also therapies that are symptomatic, that make people feel better today. We need to be able to diagnose MSA sooner, and understand what are the problems faced by patients with MSA and how these change over time. So far, we have 22 investigator teams that are, again, collaborating with us and sending their information from their patients. You can see in the US, Europe, Australia, and also in South America. These are the names of the investigators. I'm not going to read this off, but you will see there are giants in the field of MSA that's here, including Dr. Wenning, Dr. Lowe, Dr. Biaggioni, and others. So we're really excited to bring in this expertise and work with these people. So I told you we had these academic clinicians, our industry partners, and advocacy, and we need to seize the opportunity to build on the collaboration we have and make this work for us. In terms of recruitment, let me go now into some core details about the study. In terms of recruitment, we're doing well. And I'm also going to show you a breakdown of the diagnosis so you can see that this study is heavily focused on MSA. I told you that our partnership with the European, and this is enrollment, these are the number of patients we have in this study since we rolled it out. These are cumulative in the dark gray, and in the light gray, you'll see that these are our annual accrual rates. And you can see with the expansion of the study to include the support of the MSA Coalition and also the European MSA Study Group, we had a much greater increase, so that really helped us. We have so far 741 patients enrolled in the study. It's active enrollment. Our highest enrollment rates were last year when we had 170 new cases. 12% of our patients have been followed through death, so we are really capturing all stages of the disease. We have some patients that have been in the study now with 7 years of follow-up. We have implemented strategies at our investigator teams to make sure we're retaining patients, we're keeping them in the study, and we're giving our investigator teams the tools to follow people with MSA over time. And this would not have been possible without the foresight and the vision of the MSA Coalition, including, of course, Pam Bauer. This is a breakdown, this pie chart here is a breakdown of the type of patients that are enrolled in the natural history study. And you can see that the substantial portion of patients are actually patients with MSA. We are following a substantial portion of people that we believe are at high risk of developing MSA. And then we're also following a group of active controls that include Parkinson's disease and dementia with Lewy bodies, which are often diseases that mimic MSA or are confused with MSA, which is why we think there's value in following those patients along with a diagnosed MSA cohort. So let me tell you quickly about the is being used. In terms of our initial goals, we had a study that I spoke about last year in which we followed patients with autonomic failure, and we saw that those that present with autonomic failure with falls in blood pressure and also bladder problems, who have an earlier age of onset in the mid-50s, who have some signs that the nerves outside the body are largely spared, and who have some other clinical characteristics may be at high risk of developing MSA. And we've also provided a validated diagnostic test to be able to differentiate what are falls in blood pressure due to a problem with the nervous system from falls that are secondary to other medical complications, which again is helping us narrow down in where our potential pool of MSA patients could come from. How else is the data from the natural history study being used? In important ways. It's been used to help with providing information that helps shape the new diagnostic guidelines that the research community and Dr. Wenning is working on. It's been used to help develop and refine better outcome measures. It's been used for us to be able to find tools that help us get your information, for you to tell us what's bothering you over time, and to have strong, sensitive tools to see how our interventions of all those different kinds of therapies are impacting your lives with MSA. It's used to be able to support biomarker initiatives. We're working with some of the giants that Dr. Wenning mentioned in his talk to develop blood tests for MSA. We're working with Dr. Sonia Scholz at the NIH on collecting genetic information to see if we can pick up genetic characteristics that might predispose people to MSA. So all that important work is going on, but this natural history study provides a backbone for those projects. And it's, of course, aiding us in clinical trial readiness. What are we looking for? Who are the target population that we need to be focusing on for drug trials? So it's really providing a lot of information that supports this clinical development plan we have. And it provides this bridge between academic institutes, between industry partners, and in advocacy groups. So we are moving forward. We are helping with clinical trials. We're sharing our data samples. We're helping other researchers. We're using this benchmark information to help drug development and drug approval processes. And we're helping to plan clinical trials that have the best chance of success so that we can hopefully prove that these drugs we want to try can slow down the progression and we can get them approved faster. So let me just close by telling you what you can do now. And I'm going to echo Dr. Claassen's message that really we need you to participate. Anybody with multiple system atrophy can participate in the natural history study at an approved site. If you're in the natural history study, you can still be in an interventional study trial. It doesn't stop you from doing that. You just need to tell us, be honest with us, because we want to capture that information too. Stay connected, stay informed, know about what research opportunities exist, and include and encourage your friends to participate. Now, I just want to close by telling you that I fundamentally believe that an importance of the natural history study and the work we were doing is to support the careers of young investigators. We need to bring in people. We need to bring in new minds, bright ideas, and show people that MSA is a disease that has unmet needs and you can really make a difference. We've enabled access to the natural history data set to young investigators so that it can help them with their research hypotheses. And we're providing young investigators access to a steering committee to support their research work so to help them with their ideas and their publications, which are important for their not only for you but for their career and keeping them in the field and active in MSA. But we need you to share your story. We need you to, when you encounter these young people in the clinic that are studying MSA through you, we need you to share your story with those people, those young physicians in the clinic, whether they be neurologists, urologists, sleep doctors, autonomic doctors, movement disorders doctors, or ear, nose, and throat people. We need you to be able to, what did you start with? I mean, we need to put in, you need to tell your story, so that when they listen, they know next time they encounter patients who started off acting out dreams, or maybe and had a few balance problems, and then they were fainting after meals, they developed urinary urgency, and they had other clinical problems. We need that story to indicate to that young person that this is the potential, that it could be MSA. And your role in that, and your message in that, and your working with these young people and these investigator teams is fundamental for helping us pick up MSA in the earlier stages. So I want to thank you very much, and particularly thank the MSA Coalition for their support and faith in this project. Thank you very much.
Thank you so much, Lucy. Our next topic is clinical pathological research and brain banking for MSA. Dr. Dennis Dickson has devoted his career to the neuropathology of degenerative brain disease. He is a neuropathologist for the Mayo Clinic Alzheimer's Disease Research Center, the APDA Parkinson's Disease Center, as well as the State of Florida Alzheimer's Disease Initiative. He was one of the first to realize that Lewy body dementia was a common cause of dementia in the elderly, and one of the first to describe pervasive alpha-synuclein pathology in multiple system atrophy. Please help me welcome Dr. Dennis Dickson.
Okay, it's my pleasure to give you a little bit of information about the brain bank that we operate at the Mayo Clinic in Jacksonville, Florida. And if we could have the first slide, or do I control it here? Maybe I can control it. The touchpad.
It should be loaded. Yeah, I'm not good at these things.
The touchpad's
It's not moving.
working. There we go.
Did we get it? Where'd it go?
Let's just do escape.
Okay. Yep, that's not responding either.
Oops, not that.
No, that won't let it.
Oh, that's not working either.
No. All right.
Okay.
So, there we go.
No, fine.
Yep. So that one's done. That can go. And yours should be loaded. They were all loaded this morning. See, I wonder these Claassen's, I don't think they're got MSA in sleep.
Is it there?
What if we?
I do have it on a...
Okay, why don't you do that, because I think that might be faster. They were all loaded, I promise you, they were all loaded this morning. I swear.
Little technical difficulties.
Sorry. Okay. Yeah. And your folder should pop up, and there you go, right?
Do you want?
Is it? All right, sorry about that.
No problem.
Some people are saying,
Okay.
so make sure you give it some power. Thank you.
Okay, so our brain bank is in Jacksonville, Florida, and it is based in this building at the Mayo Clinic. It's the Birdsall Medical Research Building. And so I was recruited in 1997 to the Mayo Clinic from Albert Einstein College of Medicine in New York to set up a brain bank. And that sign is totally wrong. The brains that we get from individuals that make a donation are invaluable. You can't even put a price tag on it. But just a little bit about brain banking that may or may not know the details here, but individuals make a decision, the family or often the individual themselves decide that they want to donate their brain for medical research. And through a process of working with either a medical examiner or a pathologist, tissue procurement organization, the brain is actually removed from the skull of the deceased individual. To be most valuable for research, that's done quickly after the time of passing. And part of the brain is preserved, and that preserved part is used for diagnostic studies, and the other part is frozen in as cold a freezer as possible so that that can be used for research studies. But before anything can be done with the frozen part of the tissue for research, we need to know what the actual diagnosis was. And that involves a procedure that's illustrated here, what we call a brain cutting conference. And our brain cutting conference is actually a working conference, but it's also a teaching conference. The medical records are reviewed on the patients, the brain is evaluated, it's photographed, it's dissected. This is usually attended by medical residents, trainees, medical students, research trainees, and everybody gets to learn a little bit about that individual and gets to see what transpired in terms of the brain pathology. We've been doing this for over 20 years, and just in terms of MSA brain banking, the brain bank was never set up to be specifically focused to collect MSA cases, but through the years and actually prior to about 2008, we were getting about 4 cases per year, which it's not a large number of cases. But then in 2005, Cure PSP, one of the original supporters for our efforts in brain banking atypical Parkinsonian disorders, made a decision at an executive level, and I don't know how they made this decision, but they decided that it was important to actually support collection of MSA cases. And since then, we've been getting over 10 cases, sometimes more than 20 MSA brains per year. And we currently have over 270 brains of pathologically confirmed MSA. So what about the MSA that's actually, we know it's MSA with absolute certainty because we've evaluated the tissue. It's a disorder that affects men and women about equally. In our brain bank, and this reflects the nature of brain banking in the United States, and it's unfortunate that we don't have more involvement of minorities. So 95% of the individuals that decide to do brain donation are Caucasian. There are many reasons for this, and we really don't have time to discuss it, but there's a legacy of minorities being victims of misuse in terms of clinical studies, and so there's reluctance to participate in research. The average age at death is 66 years, but there's a wide age range. We have an MSA patient that passed away at age 37, and another one passed away at 91. The average disease duration is also quite variable, from as short as a year or two to as many as almost 20 years. If you look at the brains with MSA pathology, how many of them actually were thought to be MSA while that person was alive? More than 3/4, or about 3/4 of them actually were thought to have MSA. So the MSA pathology really drives a very distinctive clinical syndrome that is recognized by the overwhelming majority of clinicians. If you look at it the other way, of patients that while they were alive thought to have MSA, how many of them actually have MSA at autopsy? It's about 60%. So it means there are some other disorders that can masquerade as MSA, and they actually are PSP and Lewy body disease, or Lewy body Parkinson's. One thing that is interesting, if we think about most of the other neurodegenerative disorders, for example, Alzheimer's disease or Lewy body dementia, there's a significant proportion of patients that have a family history of other members of the family, the extended family, that might have had either dementia or Parkinsonism. But that's not the case for MSA. MSA does not seem to be a hereditary disorder. If there's a hereditary component, it's a very minor component. And so that really leaves us to the question of what besides bad luck determined why that person got the disease? And we don't really know. So where do we get the brains? They really come from almost every state in the Union. And because, and you'll hear a little bit later when we talk about the Brain Support Network, that where that group was based out of the Bay Area in California. California is actually the major source for brain donations to the brain bank in Florida. And then Florida obviously would be the next. So what happens after we do that brain dissection? So we take samples from very specific areas that we capture the most important areas that are affected by the disease, but we also take areas that are unaffected. The tissue samples that we sample are actually placed in paraffin wax. This is actually a section of the midbrain. And those paraffin sections are cut with very thin slices, about 5 microns thick, so it's thin enough that light actually passes through them. The tissue sections on the mounted slides are stained, and then the stained slides are provided to me to evaluate. We look at the tissue, the brain sections, under regular light microscopes, but also with fluorescent microscopy. We collect quantitative information about what we see under the microscope. That quantitative information and qualitative information is recorded in a written report, and that we generate a final diagnosis. It was MSA, it wasn't MSA. It was MSA with something else. It was MSA maybe with a stroke, MSA with some Alzheimer or aging pathology. That final diagnosis and the report is then entered into a database. It also is sent to the next of kin as well as to the pathologist that sent us the tissue. So, and then finally, the select cases are actually presented in clinical pathology conferences. These are conferences where we present the whole picture. We do the clinical information, the imaging studies if they're available. We discuss the pathology. We show the gross, the images of the brain, the entire brain, the dissected brain, and then look at microscopic slides. And then through a process of learning and discussion, the clinicians, the students, the residents, we learn something about MSA. And so you may wonder what the 3 shields of the Mayo Clinic stand for. They stand for patient care, research, and education. And in fact, the brain donation contributes to all of those. Not so much patient care, but in terms of the diagnosis, actually more helpful to, obviously not helpful at all to the individual, but it's helpful to the family. So I think that kind of fits under the patient care shield. It's definitely valuable for research, and it's useful for education. One of the things that we can do with brain tissue is we can look at it with an electron microscope. This is a really high-powered microscope that uses electrons rather than light to evaluate the tissue. The electron beams pass through the tissue and basically hit a photographic film or a photosensitive plate that it then records the image. And it's through methods like this we're actually able to identify synuclein molecules in the individual cell. So the other thing that is done in terms of research is the frozen tissue is used for a variety of different types of research: molecular biology, biochemistry, and genetics. And to do that, small portions of the brain tissue are ground up or homogenized with a blender, and then various components of the tissue are extracted and used for research: proteins, lipids, nucleic acids, RNA, DNA. And that allows us to actually look at the chemical changes that occur in synuclein in the brain. And the DNA that is extracted can be used for a variety of genetic studies, both that are done at Mayo Clinic, but also we participate in a number of national and international studies on genetics of MSA. So just what does the brain look like in a typical MSA? This is an MSAP, a patient with striatonigral degeneration. And what it illustrates is that the 2 characteristic features are degeneration of the striatum, and that the striatum includes the caudate nucleus and the putamen. And the arrow on the right is pointing to the putamen. That putamen is about a quarter of the size that it should be because the neurons have been lost. And also, if you notice that it has the color is different. It's a little bit browner than the rest of the brain around it. That brown color actually is because there's excessive accumulation of iron. And I think earlier this morning we talked about how imaging methods can actually detect iron and monitor the increased iron. And the arrow on the left is then pointing to the substantia nigra. That should have black pigment, and it doesn't look black at all. It looks kind of tan or light brown. That's reflecting the severe neuronal loss in the nigra. So those are the 2 areas, nigrostriatal degeneration. Many patients with MSA, that's the major pathology. There's very little other pathology that you can see with the naked eye. The other form is olivopontocerebellar degeneration. And in this form, the degeneration affects different parts of the brain. It affects the brainstem, especially the pons and the medulla, and it affects the cerebellum. And I try to do this without advancing it. Maybe if the pointer will work, I can try to. Yes, okay. So this is the cerebrum. I think actually Nick actually showed you an MR of a brain that was cut almost in this same plane. We're looking right, as if you split the brain right in half into right and halves of the brain. And this is the midbrain. This is where those substantia nigra dopaminergic neurons are. It looks pretty normal, but the pons should be 2 to 3 times the size of this. It's markedly atrophied in MSA. And then, so this is the pons. The midbrain the pons usually twice as large as the midbrain, and it's just the reverse. The midbrain's almost as large. So very severe pontocerebellar atrophy. And the pathways that project into the pons or into the cerebellum also undergo atrophy. So this is a normal projection fiber from the pons into the cerebellum. And then here's the MSA in comparison. So very discrete pathology. The thing that links these two disorders together, even though they look very different, and clinically they're very different, is the fact that they have the same molecular pathology, and that's the abnormal alpha-synuclein. So in terms of the brain bank, patients are classified as either this pure OPCA or the pure stratonigrel, and then there's a group of patients who have the full house. They have both. They're really unfortunate. Are there any characteristics that distinguish the stratonigrel from the OPCA, the MSAP and the MSAC? There are not a lot of differences. One of the things that comes up is OPCA is more common in men, and I think Nick also presented this from larger clinical studies. And that for reasons that are unclear, the MSA cerebellar form is more common in men than women. And the other thing that seems to be recurrent, even though we don't have a lot of non-Caucasians, the non-Caucasians are overrepresented in the MSAC. And in fact, in Asia, there's a really nice study from a Japanese investigator working in the Queen Square Brain Bank who also evaluated MSA patients in Japan. And MSAC is much more common in Japan, whereas in the Queen Square Brain Bank in England, it's MSAP. So stratonigrel degeneration in European Caucasians, and in Asians, it's more C. We don't know. That might suggest genetic underpinnings, but we don't have really good evidence of that. So the major pathology that unites the MSAP and MSAC is the alpha-synuclein pathology. Alpha-synuclein was discovered as the gene that caused familial Parkinson's. It was actually discovered in 1997, and a number of groups shortly after that said, well, let's look at our brain bank and see what else besides Parkinson's might have alpha-synuclein. We actually did this. We were one of the first to actually report in 1999, teaming up with Sid Gilman and his team at the University of Michigan. We didn't have very many MSA cases at the time, and Sid and Roger Albin, they sent us MSA cases from the University of Michigan so we could confirm this finding in a larger cohort. And in fact, both the glial inclusions and the neuronal inclusions were described. And using markers that were specific for oligodendroglia, we could show that in fact the inclusions, the synuclein inclusions were in oligodendroglial cells. In that same study, we actually looked at changes in the solubility. The synuclein that deposits, synuclein is not normally a lump in a cell. It's a soluble protein, has dynamic properties. It's very important for the normal synaptic function of neurons. What it's doing in oligos is another question, and we really don't have a clue. But it's present in both. And that fact that it aggregates, it tells us something's abnormal with that protein because it actually changes in its solubility. And looking at more stringent solubility, going from very soluble to S5, there's really insoluble, like it's material that actually precipitates. And you'll see that in MSA, it's very different from what you see in normal controls and Alzheimer's. The other thing, using our electron microscope, we're actually able to look at individual cells that have those synuclein aggregates and show that the synuclein, which normally you can't see in a cell, it's just diffused throughout the cytoplasm of the cell. In MSA, it actually accumulates into actual filamentous or structures that you can actually visualize. Now, I mentioned that we've submitted a number of our MSA samples to a number of genetic studies, and both conducted at Mayo Clinic by our geneticists, led by Matt Farrer and Owen Ross, and also to international studies that, especially work done by Sonia Schultz at the NIH with Andy Singleton, also at the NIH. And the bottom line from all these studies, negative. They're all negative studies. All the genetic studies have been done on MSA are essentially negative. And that fits with why we don't see a family history of MSA. This is probably not a disorder that is strongly driven by genetics. We need to go outside of genetics and think outside the box in terms of what actually causes this. And we need a lot more work on that in that area. One of the things that has been that we've used our brain bank for is to do clinical pathologic studies where we can review the medical records of the patients, look at the pathology, and try to understand why did this patient have pathology of MSA, but when they were alive, they were thought to have Parkinson's disease or thought to have progressive supranuclear palsy. And these are studies that have been led by a number of research fellows. I would just point out that, did I advance? Okay, I didn't advance. Okay, these are not quite the latest slides. I just had another version of this that actually pointed out that Shunsky Koga has actually spearheaded a lot of our clinical pathologic studies. And this paper in neurology is an important study because it says that sometimes patients that are clinically thought to have MSA don't have MSA. They might have Parkinson's disease or Lewy body dementia. And the most common reason that Parkinson patients or Lewy body dementia patients are thought to have MSA is because they have autonomic dysfunction. So a Parkinson patient with autonomic dysfunction can get misdiagnosed as MSA. And PSP patients can get diagnosed as MSA if they have cerebellar involvement. So PSP is an atypical Parkinsonian disorder that usually leads to early falls, axial rigidity, and impairment of vertical eye movements. But some of these patients have more cerebellar involvement, so they have a clumsiness or ataxia. Those patients with Parkinsonism and that ataxia, that leads them to get misdiagnosed as MSA. So how to do a brain donation? If you make that important decision, it's a difficult decision. It has to be something that you discuss amongst yourselves, your family, friends, maybe people that have gone through this, families that have gone through this process. There are some ways to reach out. You can find information at the Mayo Clinic. The Multiple System Atrophy Coalition actually has a link to information about brain donation. And the MSA Coalition has teamed up with the Brain Support Network. This is, I mentioned, is a not-for-profit organization that grew out of a family support group in the Bay Area in San Francisco. And now they've been assisting families in making brain donations, and now they've, led by Robin Riddle, they've actually got a really efficient team, a very good database of pathologists and pathologist assistants and medical examiners, tissue procurement organizations throughout the entire country that can help families if they make the decision they want to have a brain donation. How do they actually go about doing that? The other place where you can find information about the brain donation is PSP. I mentioned PSP. PSP is a tauopathy. It's very different from MSA. Some patients get misdiagnosed. But I credit our boost in terms of our brain banking for MSA on Cure PSP because they actually went out of their way to say that we should be actually engaging the MSA as well in terms of offering brain banking for this also rare atypical Parkinsonian disorder. And then finally, just point out some of the people in the lab, past and present, that have contributed to the studies, the research study, and I highlight Dr. Koga because it's been a major focus of his research on clinical pathologic and genetic studies on on MSA. And I'll take some questions if we have time. Are we doing it, God? Okay.
Thank you, Dr. Dickson, for that very informative talk. So right now, we're going to take our 15-minute break before we come back to hear from Dr. Khurana with the future directions for MSA-funded research. And then following his talk, we'll have a Q&A with the research panel. So please, 15 minutes, be back here at 3:40. Thanks. And there's snacks outside the doors.
No, we have the panel. So we have to come back at 15.
Well, because we're looking forward.